In this paper, we studied interstitial cancer migration. In order to do that, we used 3D printed structures which mimic the pores of human tissue. Through our system, for the first time, we were able to link the cell cycle and the ability of cells to migrate through small pores. During cancer metastasis, cells move from a primary tumor to a distant organ where they form a new colony. In order to do that, the cancer cells have to migrate through the dense tissue into the blood circulation, which they can use to travel to a new spot on the body. So learning more about the cellular and molecular tricks that cancer cells use to invade the tissues is very important because it can lead to improved treatment strategies. Despite the importance of metastasis in the cancer progression, there are still some key factors of this process which are largely unknown. One of them is the role of the cell shape adaptation when cancer invades dense tissues. So our goal was to make a platform with high spatial and temporal resolution which mimics the resistance of tissue to cancer migration. Efficient pore penetration is the result of the interplay of multiple factors related to the cell and its microenvironment. We investigated the role of cell deformability, cell adhesions, and DNA condensation. We demonstrated that during the first phases after cell division, the synergy of those critical biomechanical characteristics contributed to a more invasive cell phenotype. In early G1 phase, the cells have still not established mature adhesions, but they exhibit very high deformability, while in G1 phase, the decrease of deformability is compensated by the increase in focal adhesions. So the small pores existing in dense interstitial tissues such as the human dermis may act as a cell cycle phase filter which blocks the dissemination of individually migrating cancer cells until a division occurs. We used the microfabrication method which was developed in LTNT by previous members of the lab, called 3D Electro-Hydrodynamic printing. We used colloidal nanoparticle ink to create arc-like structures on a biocompatible polymer substrate with ridges. With this nanoprinting method, we were able to produce pores with very controlled geometry and shape, with cross-sections that range from a few microns to hundreds of microns, so in the physiological size range of the pores in the human dermis. We use substrate with ridges, which promote cell directional migration, and HeLa cancer cells and breast cancer cells, both with fluorescent nuclei.
So we have developed a unique technology that allows us to make unprecedented experiments in cancer migration. And by doing that, we have demonstrated that there is a connection, a functional link between the ability of cells to migrate in complex environments which are very dense and provide small passages to cells, and their ability to replicate. Of course, when we think about these two capabilities in a cancer population, it is natural to think that this might be hijacked by cancer cells in order to invade tissues and disseminate. So altogether, we have new concepts toward the understanding of how cancer cells may become metastatic, may invade different and distant tissues, and generate secondary tumors.
Our results support a model in which a population of malignant cancer cells that divide more often or stay more in phases soon after division may penetrate interstitial tissues faster and with higher efficiency. This finding is very important, especially in relation to drugs that arrest cells in exactly that cell cycle phase, which we found to be more permissive.